Hello, hello. Welcome, everyone. I am Kevin Curran, and I am sharing a series of videos related to the delivery of gene editing cargo, gene editing medicine into the human body. These videos are just an experiment. I'm seeing if this is of use to people. I'm sharing them on YouTube. These are excerpts from courses, lectures that I am delivering to different biotech, pharma companies, nonprofit, government groups that are interested in advanced medicine. And I'm putting some of this material up for free as a educational resource on YouTube. So if this is helpful for you, subscribe, share, and I'll keep creating these little video excerpts. The topic I'm focusing on right now, I think is critical for the future of advanced medicine, durable cures for diseases. We have the capacity to precisely edit DNA, but now we need to figure out how to administer it into the human body so that these tools, whether it's CRISPR-Cas9 or future iteration of gene editing tools, possibly prime or base editing, tools, can get to the right cell types in the human body. You want to think about disease phenotypes. Where does the disease manifest in the body? Usually it's one particular cell type. Well, we can inject CRISPR-Cas9 into the bloodstream, but that doesn't guarantee safe passage to the kidney or the spleen or solid tumors, glioblastomas inside the brain. We need to assist this cargo with some sort of a delivery strategy. Okay, so this is a big focus of biotech companies right now. So I think everyone's familiar with gene editing. This is the benefit. We can precisely edit human DNA on the chromosomes inside the nucleus of cells, but we've got to get the cargo to the right cell types. So this image is from the Jennifer Doudna's lab. This is a recent review this year, 2024. The image shows sort of a roadmap of all these tissues in the body that we're trying to get to. And I'll show in a second tissues we are successfully reaching. But you can see, here we've got a couple delivery options. One is just what I call naked cargo, just taking the CRISPR-Cas9 either in, two RNAs, RNA for the guide RNA and RNA for the Cas9 enzyme, and injecting that into the body. That can be done in an ex vivo context if we remove cells from the body, right? So I'll talk about that. It's possible we can deliver that directly into the bloodstream for some diseases, or maybe localized delivery. So I'll talk about just naked cargo, sort of delivery that doesn't rely on a delivery system, a vesicle. Then I'll talk about lipid nanoparticles, a synthetic ball of fat, and that's proven useful, especially for delivery of gene editing to the liver. It's a challenge to get LNPs to deliver outside of the liver. And I'll talk about the newest research on a different type of vesicle called a virus-like particle. And the idea here is we're going to take elements from viral vectors, viruses, and we're going to kind of remove any safety issues and then sort of just use the outer coating of viruses to deliver cargo. So that's a lot to talk about. I'm going to break this up into 3 videos. Today, I'm going to shoot for less than 10 minutes, and I'm going to talk about using viral vectors, AAVs, adeno-associated viruses, and lentiviruses. And I'll talk about naked cargo, what's happening, what can be done with just delivering the gene editing cargo, right? The CRISPR-Cas9. And then in subsequent videos, I'll summarize the state of research with LNPs and then virus-like particles. All right. So, I I think we appreciate this. Diseases manifest in one part of the body. So it's an issue of delivering cargo to the right cell type with specificity. And so you need to bring sufficient volume, enough concentration of the gene editing tools to engage with the cells to make a meaningful shift in the cell's genetic programming, right? So we don't need to just reach, say, the kidney cells. We need to reach enough kidney cells that we're editing enough population of those cells to actually transform the disease and eliminate the symptoms of the disease, hopefully forever, right? That's the promise of gene editing, is this durable treatment. So we think in terms of gene editing efficiency, delivering to the cells, and then what's the efficiency? What's the rate of altering those cells? And then is that going to be meaningful in terms of what really matters, which is, the quality of life for the patient? Are we actually knocking out, the clinical symptoms of the disease? And we want to minimize off-target hitting. We don't want to be editing cells, the non-disease cells. So we want, Cas9 is it's an enzyme that chops DNA, either a double-stranded break or a single-stranded break. Either way, we don't want that Cas9 nuclease chopping up DNA in unintended cells. That's going to increase the risk of safety issues, adverse events. So it's really important to be able to precisely bring gene editing tools into the right cells at the right volume. You get the right genetic editing efficiency while minimizing off-target effects. Okay, so the good news is we're 10 years into doing this, right? The Doudna-Charpentier group, they made this tool programmable in about 2012, and now it's moved into human clinical trials. It's 10 years later, December 2023. A company named Vertex, they partnered with Crispr. They got the first gene editing drug approved for sickle cell disease. This drug is called Casgevy. It's now, working its way into the patient population, which is great. Medicaid is figuring out how to finance this, how to reimburse. That's important. But what I'm interested, in these videos is the delivery. So what's interesting is that this first approved gene editing drug was for ex vivo delivery. So that tells you, we haven't figured out how to inject this drug into the body yet. In order to deliver it safely with potency, we have to remove bone marrow stem cells from the human body, get them in a lab, and then we can apply CRISPR-Cas9 directly to these cells where we have total control, and we can dial in the concentrations to get great efficiency. So that's what Vertex is doing, and I'm going to talk about that in some detail because they're using electroporation as the delivery. They're using essentially a blast of voltage of electricity that loosens up the cell membranes of the bone marrow stem cells, and CRISPR-Cas9 slides in through the membranes into the nucleus and then edits the patient's hemoglobin gene, right? We're talking about sickle cell disease. So there's a hemoglobin mutation that needs to be addressed. So it's in that case, electricity is the delivery system. Now, obviously, that's not going to work for cells that are deep in the human body that we can't remove and move into a lab, right? So how are we doing in this pursuit of in vivo delivery, actually bringing CRISPR-Cas9 into the body and then using a delivery system to bring the tools to the right cells? Well, I'll mention Intellia. Intellia, to the best of my knowledge, they are the farthest along clinically. They just initiated a phase 3 trial. This is a pivotal trial. If it goes well, they should get approved for a liver disease called ATTR. It's a type of amyloidosis. So Intellia is trying to knock out a toxic gene. They're trying to eliminate the TTR, so they're using CRISPR-Cas9 to bind a TTR and deliver some mutations, some indels, insertion deletions to knock it out. If that works, you get an improvement in this patient group. So how are they doing this? They're using lipid nanoparticles, right? The same type of lipid nanoparticles that Pfizer and Moderna used for their COVID vaccines. All right. Interesting side note, people often ask, if we were able to use LNPs for the vaccine, why is this still a challenge for gene editing delivery? With the vaccine, we only needed localized expression of RNA. So the LNPs in the COVID vaccines, they held an mRNA for the spike protein that hit muscle cells and APCs, immune cells, in a localized manner. But that was sufficient to instigate immune memory because these cells presented the viral spike protein to circulating B and T cells, and then we got the systemic immune memory. So that LNPs do work well for in a localized context into muscles and antigen presenting cells. So that's great. You can think of that as one successful administration of cargo, RNA cargo. This is slightly different, but same principles. We're trying to deliver guide RNA and then either Cas9 RNA or Cas9 protein. I'll talk about that in a second. But Intellia is making progress here. And it's notable that they're hitting the liver cells. So speaking of LNPs, LNPs are one of the most promising delivery vehicles for gene editing cargo, and they seem exceptionally well suited for liver delivery. So if there's a disease in the liver cells, that there's a good chance we can get CRISPR-Cas9 into those cells. Why? Why is that easy? Why is that? You can think of it as the default mode for LNPs once you hit the bloodstream. Well, the liver organ filters out blood. So if you bring a drug like gene editing tools into the bloodstream, they're going to pile up on liver cells. They'll accumulate there just as a function of the liver purifying blood. That's one piece, sort of, I guess the systemic sort of mechanical end of that delivery. There's another element that isn't discussed as much, which is lipid nanoparticles are synthetic balls of fat. Well, when they enter into our body, an endogenous, a natural protein that exists in our body called ApoE will accumulate on the surface of LNPs, just because ApoE, it's a protein that binds to fat cells like lipids. ApoE will specifically bind to liver cells, to the LDL receptors on liver cells. So that's sort of this natural inclination for LNPs to form a cell-specific bind to liver cells, and then the LNP, once bound, they then will climb inside. They'll enter in to the liver cells through endocytosis, and then they release the CRISPR-Cas9 right into the cytoplasm, and then the guide RNA will bind up complex with Cas9 protein and enter the nucleus to make an edit, in the case of Intellia, to the TTR gene. All right. So LNPs work well for liver. One of the big challenges is trying to get LNPs to bind outside of the liver, right? It's almost to move it off of that default mode of just going from bloodstream to livers. All right. So those are, I guess, two of the biggest success stories today in 2024. It's the ex vivo delivery to bone marrow stem cells, which then, develop into red blood cells, so that's a good use for sickle cell, and then the Intellia piece for liver cells. So when we think more broadly than that, what does good look like? What are our objectives here with gene editing delivery? We want to be highly cell specific. I told you why. You need high efficiency to the target cells and and then you don't want to be entering into non-target cells. You also want transient delivery, and this piece you don't hear discussed that much either. Depending on the disease, it may be optimal to just have a short time window where that Cas9 enzyme is doing its work, is actually chopping DNA to disrupt a toxic gene or to insert repair template, right? That it could be just maybe a day or maybe 12 hours or 48 hours where you want that actively occurring. And then you want the Cas9 system to shut down, right? Because you don't want it to keep chopping up DNA. It's going to cause problems. So there's a high value to transient delivery, or what we think of as like tight temporal control, tight control on that time window when the editing actually happens. That seems to be a big objective with a lot of research groups right now for the reasons I just mentioned. And as I'll explain in just a minute, that's why the use of viruses, viral vectors, may not be ideal, because AAVs and lentiviruses they could increase that time frame of when Cas9 is active, and that could cause safety issues. So that is a concern. And then the one other piece, which I've sort of hinted at, but ultimately, if we want to really hit, 7,000 genetic disorders in the world, right? If we want to hit all these and move broadly into patient populations, not just in wealthy nations, but in low and middle income nations, we have to think about how expensive it is and how complicated it's going to be to administer to this program, this therapy to each patient. And an ex vivo therapy is always going to be more complex and expensive than an in vivo. So ultimately, we want to figure out how to just be able to directly inject into the bloodstream, a one and done injection in a blood clinic, something like this, something that doesn't require a high-end medical center like a Stanford or an MD Anderson. That's not realistic. When you think globally. So there, there tends to be cheaper manufacturing for in vivo, cheaper medical costs, and less logistics if you can go in vivo. All right, I'm generalizing there, but so there is an impetus to design systemic in vivo injections that are still cell specific. All right, plowing forward, we should start by just discussing viral vectors. This is sort of we have many decades now of using viruses, modified safe viruses, to make genetic modifications, right? So that there's a lot of research here that we can rely on. We've been using a lot of work. I'm not going to go into the whole history of it, but Novartis got Zolgensma approved. That was the first AAV gene therapy approved in the U.S. And that was in 2019. We've been using lentiviruses for many therapies now. The whole CAR T industry really relies on lentiviral constructs. So we can take these viruses and sort of pull out the harmful viral component and then rely on the high efficiency of viral vectors to transform cells genetically. But we can just reapply that with gene editing tools instead of these more traditional gene therapies. So the benefit in general to using viral vectors for gene editing cargo is we're building on science that already exists. We're building on delivery systems that have been used and have been scrutinized for a long time. And all that science is building on hundreds of millions of years of viral evolution. Viruses had to learn how to dock up on human cells. So they have they have this machinery, this cell-specific machinery already sort of figured out through evolution, right? AV relies on, what we call serotypes, cell tropism, to bind to cells and then climb inside and deliver genetic tools. And lentivirus does as well with their own kind of glycoproteins and whatnot. So the benefit to using a lentiviral vector, like I said, there's a whole industry that pumps these out for the CAR T cancer therapies. They can carry a huge amount of cargo. Lentiviral vectors can carry about 10 kb, 10,000 bases into a cell. That's important because when you think of all of the genetics that we need when we bring in guide RNA, Cas9 RNA, it's somewhere in the range of 6 to 7 kb, right? In general, this will probably change quickly over time, but right now, a lentivirus would work in that context, as opposed to the other commonly used viral vector, which is AAV. That tops out at 4.7 kb. So that would be tricky for gene editing delivery. And like I said, we already know how to design how to manufacture lentiviral vectors. They're pseudotyped, which means we already have quite a bit of cell-specific equipment here, cell-specific promoters. So a lot of that cell transduction machinery is already in place. AAVs, similar story. The tropism, tissue promoters, cell specifics, that's good. Now, what are the downsides? I already hinted at that in that viral vector expression, it may not be ideal for transient, quick delivery where we quickly edit a cell population, maybe a stem cell population. We make the edits to the chromosomes, then all the downstream cell divisions off of that edited population will carry the genetic fix, right? That's great. And so that may only require a tight time window where Cas9 enzyme is actively cutting DNA. And so a viral vector, it may be too much Cas9 activity. It may be too much gene editing, right? Too long of a window there. It's not a transient approach. We don't want to overexpress Cas9 nucleus. Without going into details, the lentiviral vector will integrate cargo into the chromosomes, and so I don't think we want that. I don't think we want Cas9 integrated into a chromosome because then cells will just keep expressing it. So I know some groups are working on kind of eliminating that piece, but that's going to be an issue. And to a lesser degree with AAV. AAV doesn't integrate into chromosomes for the most part. They have an episome, but still that may offer too much chopping activity. So those are the pros and cons of viral vectors. I am just looking at the time and realizing I aimed for 10 minutes. I think I'm already at around 20 minutes. So there you go. I'm Irish. I've got the gift of the gab. It's a blessing and a curse. I'm just going to keep going. Hopefully long form content is coming back in style and people won't mind watching a longer video. Because I want to kind of wrap this up. I want to kind of put a bow on this because I started by talking about viral vectors, how there's advantages there, but it may be too long of a time window for editing to occur. And again, we have to think every disease is going to be its own strategy, right? That'll dictate the cell type, but also the length of time where we want editing. That's going to also be a function of the efficiency, the amount of cell types we're reaching. So, there's many ways to skin a cat, different tools for the job. So I'm just giving a review here. Viral vectors carry the possibility of a longer duration of gene editing to occur. If that's not desired, you can strip this all the way back and think about naked cargo. All right, gene editing tools with no vector, no LNPs. Can we just deliver guide RNA and the nuclease Cas9 into cells? The answer is yes, certainly with ex vivo and possibly with in vivo. The benefit here is this is probably the most transient. It's the tightest temporal control, because as soon as guide RNA Cas9 enters our body, they are subject to degradation through nucleases, RNases. Our body's going to chop this stuff up and get rid of it. That's why that's part of the problem. That's why we're trying to coat them in LNPs or whatnot, or vectors. But maybe a short burst of transient editing is ideal. So, you're reducing off-target editing, long-term toxicity that Cas9 might create, or insertional oncogenesis, right? Lentiviral vectors, you might integrate the Cas9 into a necessary gene, and that could trigger a problem, like a cancer in that cell population. So you kind of minimize all of this piece, because nothing's integrating into chromosomes, except the precise edit. You're not integrating the Cas9 into genomes. So this can be done in two ways, and both are being explored. You can think of one as like the dual RNA approach. So you're going to just bring guide RNA and the RNA for Cas9 into cells, and then that Cas9 will translate into proteins in the cells. And then you can also think in terms of what's called RNP, ribonucleic protein, which means in a lab, we're going to form Cas9 protein and then let it pre-complex with guide RNA. So we've got pre-complexed Cas9 and guide RNA that will be delivered into the cell. That is what Vertex is doing with Casgevy for sickle cell. Okay. Now they partnered with a company called MaxCyte. MaxCyte has this electroporation tool I touched on earlier. So they're going to electroporate RNP into bone marrow stem cells. All right. So this is naked cargo. It's just ribonucleic protein. It's Cas9 complex to guide RNA, punched in with electricity. Now, it's ex vivo, so there's a lot of steps. This is a big ask for the patients. They might be in a hospital for a few months. Their blood is apheresed, right? First, you have to mobilize their bone marrow stem cells from their bones. That's the process. Now you've got peripheral bone marrow stem cells that can be extracted through apheresis. And the cells then are brought to a manufacturing facility where they are then electroporated with the RNP complex, gene editing complex. And at this point, these are usually it's an isolated population of CD34s, bone marrow stem cells. You can do a little quality control on these, and then you invite the patient back in, and you can infuse these back into the body, and then the cells will engraft. They'll engraft back into bone marrow, and then the natural development will take over, and red blood cells will develop from this stem cell population, this genetically modified stem cell population. And now you get red blood cells that are producing hemoglobin. And this works. So let's see. Here's just an image that shows this. The Cas9 protein is complexed. It's coupled up with the guide RNA. That will happen naturally, right? Remember, CRISPR-Cas9 evolved naturally in bacteria to attack viruses. So there's a natural affinity with the Cas9 protein to bind up to guide RNA and form the complex. So it's ready to go and bind and make gene edits. So you're sort of you're sort of co-opting that natural piece of evolution. And then the blast of voltage. I've checked out the MaxCyte website. The voltage relaxes the cell membrane. I used to do stuff like this in grad school when I was making my clones, doing genetic engineering. So it's this technology's been around for a while, but apparently MaxCyte has perfected it, has gotten it quite better. So they have a relationship with Vertex. Their system is being used to manufacture these Cas9 cells. And I pulled this from the paper, the New England Journal of Medicine, that described this drug. So they're seeing 80% efficiency. So 80% of these bone marrow stem cells, once they're removed from the body, are being edited. And then once they're moved back into the body and differentiate into blood cells, they're seeing fetal hemoglobin levels increase to 29% compared to 10% in control cells. So a 19.5% increase in hemoglobin in these cells once they're back in the body, and then you see the clinical outcome that shows the real benefit for sickle cell patients. So this is phenomenal. I mean, this is groundbreaking science, and a nice option for sickle cell patients. So anyway, this is naked cargo. It's naked gene editing tools. It's just RNP complex, and then because it's a ex vivo situation, you have the benefit of using electricity. This isn't going to work in an in vivo context, right? Just imagine that. Imagine you're trying to get into the spleen. How would you apply the voltage? Where would you apply the voltage to target those cells? It doesn't seem realistic. So I think this option, it's highly transient, right? You're just bringing in the RNP. You're not integrating Cas9 into chromosomes, highly transient, but you have total access to these cells, so you can really hit a big percentage. They're hitting 80% of the cells in a dish, right? Then you can run some quality control, make sure the cells look good, and then move them back into the patients. So ex vivo has a lot of advantages. The big disadvantage, like I mentioned, is it's really expensive. There's a lot of steps, a lot of medical steps. It's a big ask for the patient. They need to keep coming back for multiple procedural steps. And it creates a lot of costs, a lot of medical costs. And in this case, it requires busulfan, a conditioning regimen, which is kind of it's a pretty harsh chemical that patients have to go through before these modified cells are infused back into their body. So again, in vivo is kind of the goal. So how can we take the win here with Cas9 and then build on that for in vivo delivery into different cells in the body? All right, I'm almost done here with this first installment on gene editing delivery. I just wanted to mention, it is possible. We are trying, the community, the biotech community is trying to deliver naked cargo, ribonucleic proteins, directly into the body, either in a systemic way, bringing it into the bloodstream, or a localized way, maybe into the eye or the ear to hit, localized cells. The problem is you get very low efficiency, very low transduction. So transduction is how many cells are we actually, bringing that RNP cargo into and then making those confirmed edits. Like electroporation would help, but that's not possible in a human body. And also, there's nothing about the RNP that's specific to any cell type. So you lose the cell specificity of the viral vectors if you go with naked cargo. So you can see a trade-off here between transient Cas9 cutting and cell specificity. So, what we're seeing is editing efficiency is correlated to the distance from the injection site. So if you inject right here, you could be editing with our naked cargo. You could be editing, say, skin cells or muscle cells really close to that injection point, because the RNP can spread over maybe 100 micrometers, something like this, like a fraction of an inch. But we're not seeing editing, on the other side of the arm. Okay, that's a quick summation of the research that I've read. So definitely not systemic, but it could work in a very localized way. Here's a paper, targeting neurons, but we were only editing, this group only edited within a radius of several hundred micrometers from the injection site. One possibility is to add some kind of a cell localization peptide or maybe glycoproteins onto the RNP, the gene editing cargo. So there's groups that are adding small proteins, small peptides to the RNP that can bind to specific cells and help them, increase the transduction efficiency into those cells. Okay, it's all very early work. Okay, I'm going to stop right here. I've walked us through the viral vector delivery system and the naked cargo in an ex vivo context with electroporation. Clearly works. It's on the market. And then the early work with trying to bring RNP complexes into the body systemically or localized. In a couple weeks, I'll make another one of these, and I'm going to talk about lipid nanoparticles and where we're at with that. And then I'll make another one a few weeks later with virus-like particles and talk about maybe pros and cons of LNPs versus virus-like particles. Again, if this is of value, please share with your community, like, subscribe, all that. And I'll keep making these as long as they're helpful, as long as people are watching these. I like to talk. I like to teach. And so I'm happy to keep making these. But let me know if this is helpful. All right, folks. I am Kevin Curran. I am out of here. Enjoy the rest of your day.